Hello, so here we are on our third and final vodcast on protein synthesis. In this particular one, we're going to look at how the messenger RNA transcript works its way to the ribosome to actually form an amino acid sequence. So, to jump right in. So the translation piece, we've already talked about transcription, the formation of RNA from the gene on the DNA. So in translation, we already have that transcribed, and now it's left the nucleus and is moving out to, any thoughts? Ready? 3, 2, 1, the ribosome. Okay, so that's where our transcript is going to head. Protein synthesis, or the translation piece of protein synthesis, can be broken down into 3 steps. First, we have the initiation. This is where the transcript is actually going to attach itself to the ribosome. And the small ribosomal subunit will first meet up with what we call the first methionine codon. Okay, so the beginnings of all of these begin with an AUG start codon. So this is how the ribosome knows where to lock on. In essence, it looks for it. So the small ribosomal subunit comes in and attaches the larger one down. So just to give you an idea and to look at. So there's the piece. Here comes my transcript out of the nucleus. It gets processed. Don't forget, processing occurs first, goes to the ribosome. You've got your first tRNA here working its way in. Then the large ribosomal subunit is going to move, come in on top, and attach once it finds, in essence, that first start codon. Once it's attached in, part 2 of translation is called elongation. And in a sense, makes sense, because we're going to elongate the amino acid chain from here. So here's basically where the magic happens. The initiator tRNA anticodon, which is the UAC, which makes sense, because remember, my AUG is the codon. Okay, on tRNA, I have that anticodon piece, that 3-base sequence that complementary pairs up to the codon on the mRNA, and of course, UAC. So that's where that comes from. It matches with that, complementary to the AUG, at the P site of the ribosome. Okay, so here it is, it comes in, okay. Then we get the second tRNA coming in, binds to the next set, or the next codon here. Here's my next codon. Okay, so if I look down, again, remembering that the tRNA, the right one, comes in because of this complementary anticodon to codon base pairing. That attaches in at the A site. Then one of the rRNA, so what the ribosome is made of, right, in the large subunit will catalyze. Whoa, what? RNA catalyzing? Sounds an awful lot like an enzyme. So the RNA, this is why we've always said most enzymes are proteins. Here is an instance where nucleic acid's actually acting like a catalyst. So it's going to catalyze the formation of the peptide bond between these 2 amino acids. So right here, and notice there's that link in the chain. Okay, it attaches right to it. So we have our first 2 matched up. Once this happens, okay, oh, there it is. Once this happens, the first tRNA, that initiator, the tRNA itself, lets go, says bye-bye. The tRNA, okay, that was in the A site now shifts to the P site. So this one here now moves over, which is that. And now this is now in the P site. A new tRNA with a new codon matching up to, new anticodon matching up, comes into the A site. Those get linked. And then that one moves over, another one's catalyzed, and it continues on and on and on until, okay, it reaches what we call the end, or in essence, the stop codon. So again, these next steps, they repeat until we reach the end. Okay, because this polypeptide chain is growing, and then it will become folded to become its protein. So the final part is called termination. Termination is when we reach the stop codon has moved into the A site. Once that stop codon has moved in, because it will not be carrying an amino acid, that in essence tells the ribosome that it's done. It will release, okay, the new polypeptide chain, and that ribosome will then go off and find itself a new transcript to work. All right, so that's it. All right, and that particular mRNA transcript can be used again. The ribosomes are used and reused. So again, remembering back to that sort of animation, my ribosome is my chef, the transcript is the recipe. So ribosomes can put together any protein as long as, you know, depending on what you feed through it. So if you feed through a particular mRNA transcript, it will build that amino acid sequence. Okay? So, used again. And then we have what we call sometimes polysomes, where we have multiple RNAs working through a single mRNA transcript at the same time. Or, yeah, multiple ribosomes. All right, so a quick summary. Here's the whole process, okay? Beginning from gene. Okay, so here's my DNA in the nucleus. It stays in the nucleus. It can't leave through the pores. It's too big. So we create the transcript. Transcript gets processed. RNA leaves, heads to the ribosome, okay, where it links in. All of the codons are read by the corresponding anticodons on the tRNA to bring in the correct amino acid sequence. And voila, we get a final protein once this has been coiled.